Well, hello everyone. Thanks a lot for taking your time today to join us for today's webinar. Uh, well, compared to the last webinar, we will have three smaller presentations that will last 15 minutes each. And after each presentation, we will have 15 minutes, 5 minutes for discussion. And today, Nuno Capela, Artur Sarmento, and Soraia Santos, who received ITC conference grants, will present their work. And I hope I pronounced the names of our presenters correctly. And now I pass the microphone to Nuno Capela to tell us something about what he's been doing.
Okay. Hi, Nemanja. Hi to everyone. So I'm Capella from the University of Coimbra. Interestingly, the other colleagues that will talk, they also work with me. And we basically, we all had the ITC conference grants for to participate in the EuroBee conference. And in there, we took posters to present our work. Let me share my screen. Yeah, I need you to
Yes.
to allow. Okay. So I'll share this second screen. Put this in presentation mode. So I think you can see the presentation, right?
Yes.
Yeah. Um, so basically the work that I took there, it was about the Acetamiprid and the experiments that we have been doing here on the team. For the ones that also were in Larissa this year in the conference. So this will be basically the same work. So, but in this case, I took it as a poster, and it's about, of course, acetamiprid. This is a neonicotinoid. Also, by the way, this is the poster, so you can see how it was organized with introduction, then all the materials and methods. And basically what I presented here, it was, you cannot see my mouse, right? You can't. Yeah, but basically every line that we see here, it's somehow a different, oh yeah, you can see it. That's good. So these are different studies that we performed in here, and I will take you through these different studies that we did. So as you know, acetamiprid is a neonicotinoid. Um, and you might heard about the neonicotinoid ban all over Europe. So basically, it was proved that these active compounds were toxic, mainly to honeybees and other insects. And there were several restrictions that were applied. And the restrictions were so harsh in most of them that then the producers did not apply to renew the active compounds. So in the end, these products were out of the market. But as you can see, there's still one that is acetamiprid that is still in the market, and it's still here until 2033. And I'm using somehow this joke of Mr. Elton John with I'm still standing, because so far this is the only neonicotinoid standing. There are new neonicotinoid-like substances in the market, but they are not called neonicotinoids. Other names like sulfoxamines. Uh, we'll get to that. So basically what we decided to evaluate was how this compound, if this compound would affect honeybees. And we decided to evaluate this in real world scenarios where acetamiprid has been applied. So in the first experiment, we had these 5 different windows in which this yellow area is the area that was sprayed. And these dots are samples that we took from the landscape. And we also took samples from the hives that were installed in the middle of the landscapes, this red dot. And the goal was to measure exposure, to know how much of the pesticide is taken into the colonies. And what we saw is that then we collected samples from the hive, samples from the trees and shrubs, mostly flowers, okay? And these are the levels in PPB that we found. So on flowers, of course, we found more acetamiprid than on fresh nectar. But the interesting thing is that these levels are quite low, and there was an overall reduction of 3 times in 15 days. So in 15 days, most of the acetamiprid had degraded up to 3 times. And one other thing is that, which is quite normal and expected, is that as we increase the area that was sprayed, the amount of contamination in nectar and in pollen has increased as well. So what reaches the colonies? But even like that, we decided to check if these levels of contamination were too high for honeybees. And basically we extrapolate the levels that we found, the highest levels that we found, to individual levels. And we saw that the exposure to toxicity ratio was very, very low compared to the trigger values set by EFSA. So these were at least 10 times lower. So this means that this level of contamination was quite low. And in this case, it was not possible to evaluate resilience because we didn't have anything to compare. We only had exposed colonies, and we measured before and after the stressor. Then we decided to do another experiment, and in this case we used control colonies and exposed colonies, because in this case we wanted to really check for effects, if there was some effects by comparing non-exposed versus exposed. We also measured the residues reaching the colonies, and in this case the exposure was so low as well, and it was like 52 times lower than the sublethal effect that we found in bibliography. And basically in here you can see, sorry about this graph, doesn't have like good quality, but basically this is like the true apiaries, these two, the blue and the red. And in this case, this is the population, and it was very similar. And also the brood nest cells, it was very similar. This was when we did the application, and we couldn't see any differences. And we also wanted to know why this the amount of residues was low. So we even evaluated the denses. So we had like an observation hive in each one of these areas, and we we have analyzed the denses to know where bees were foraging, and basically most of them were foraging outside the area that has been sprayed, because in this case it was just on the first 500 meters. And as you can see in here, this was on the same date as the spraying date. Most of the bees were foraging outside the area that was sprayed, so it's obvious that they were not exposed as much. And in here we can also see it because we had scales installed on these colonies, and the control apiary, it's the green one, so no exposure in here. This blue one was where we got the exposure, and as you can see, the weight was stable. So this means that the bees were not gathering resources at this time of year from the landscape. So of course their exposure had been so low. Then we were quite keen on seeing if there are more differences. So we were trying to find some differences, trying to find if this was really safe for bees. So we did another experiment in which we evaluated also the degradation of the residues through time. And we found residues. Also, we even measured the residues on bees, and we measured the degradation through time. And we also wanted to have exposure. And in this case, we got it. So as you can see in here, the bees were interacting with the landscape when we did the spraying. So they were collecting resources from the landscape. So there was exposure. And we even tried to see the degradation of the product. So you can see in here that one day after the spraying, you have, in this case, it was 1 ppm of residues in nectar. And then on the next day, you have the reduction and you could only find like 20% of it. So there's a huge reduction from one day to the other in nectar, which does not happen, for example, in pollen. It happens that it's very stable in the landscape, also because pollen has the ability to contain these pesticides. And we also saw it that it accumulated on the hive, on bee bread. And on bees, it was quite, the levels were quite low. And then we had this peak in here, which we found it hard to explain. So maybe there was some accumulation in the hive, and then these bees died, because these were dead bees that were in front of the colony. So as you can see, like, this is sublethal effects have been found at 5 ppm, and we did not even got that close. In here in nectar, we had 1 ppm. Then all of the other values are much higher. And in this time, we measured we had also control and exposed colonies, and we did not see any differences at all in the colony strength and number of brood cells. So we decided to go and do another experiment, this case just focusing on sublethal effects. So we controlled the exposure and we saw the effects, and it was sublethal effects. And for this, I have here a small video in which we collected bees one by one, forager bees, and we fed them individually with a small amount of pesticide, and we tested both acetamiprid and another product that is sulfoxaflor. And we installed a RFID chip on the bee. And after some minutes, so for the bees to assimilate the compound, we released the bee 1 kilometer away from home. And we wanted to evaluate how long how long it takes for the bee to reach the colony, and if it reaches the colony. So in this case, we are measuring the homing ability of the bees after being exposed to these compounds. And what we saw is that for acetamiprid, all of the bees reached the colony, and you see in here the control is this line is the control. And when we fed the bees with 32 nanograms of acetamiprid, they even managed to perform better than the control. This was not statistically different it was like 0.07. But even like that, there is this tendency, and we believe that this is somehow a hormesis effect. And we believe this happens because the acetamiprid, it acts on their brain, creating excitement. So on really low dosages, it would be like the same for us as humans taking a coffee. Of course, if you take 20 coffees, then you start to have negative effects, but only one, it helps you to perform better. And in here, probably it acts more or less in the same way. As on the other hand, for the sulfoxaflor, as you see here with 26 nanograms, so much lower than what we tested for the acetamiprid, for the 61 of acetamiprid, we have effects. So only around 20-something% of the bees reach home instead of the 80%, 80 to 70%, which is quite interesting since this sulfoxaflor is in the market, or came to be in the market somehow to substitute neonicotinoids. But at this point in Europe, it's forbidden to apply it outdoors, only indoors. But the same doesn't happen, for example, in America, in which you can apply it everywhere. So in the end, when it comes to acetamiprid, we did not find evidence of negative effects. Of course, the spraying time highly influences the colony exposure, so you can apply some mitigation measures to reduce the risk of exposure. And these new compounds, unfortunately, have even worse effect at individual level than the acetamiprid. And that's it. Thank you very much. So if you have questions, I believe you can just turn on your mics and camera and go at it.
If we don't have any questions, we can.
Hi.
We have Iveta.
Yes.
Iveta.
Okay.
Hi, Nuno.
Hello,
Thank you
Iveta.
for your presentation. It was really interesting, and especially the fact about a little dose of acetamiprid is even beneficial for bees. It was really surprising for
Don't tell
me.
this to beekeepers, please.
Yeah, I think that the other problem we have is acetamiprid. It is not so bad for bees. And is it true that you can apply a bigger dose of acetamiprid, for example, if we compare with other neonicotinoids? And I think the problem is that we have more residues in honey of acetamiprid. And it comes out that, for example, beekeepers who wants to do the bio honey, so they have a problem because you always have some little amount of residues of acetamiprid in honey. So it's like another problem for beekeepers. Maybe it's not so bad for bees, but in the end we have residues in our products.
I believe that what is happening is that since we Europe have forbidden or applied the restrictions for the other neonics, now the amount of acetamiprid that is being used has increased. So of course we are increasing the exposure, and until, let's say, they have something to substitute this, people will keep using it. But at least it seems that it's less toxic. But of course, it depends on the exposure. If we increase dramatically the exposure, then we'll have problems again, for sure.
Okay, thank you. Thank you for your presentation.
Okay.
If there are no more questions, we can have Artur go with his presentation. And also, I'm terribly sorry the meeting will end in the few minutes. I'm not sure what caused this inconvenience, and when this call breaks, just enter with the same link again.
Okay, can you hear me?
Yes.
Okay, perfect. So I will start the presentation. Can you see the first slide?
Yes.
Okay, perfect. I have a direct feedback at my side with me. So thank you. So my name is Artur Sarmento. I'm a PhD student at the University of Coimbra. And the work that I'm going to present here, it's a tiny part of a much bigger study or a much bigger project. It was EcoStack, which was a Horizon 2020 project, which we as the University of Coimbra were involved. So the title of my presentation and of my poster was Oral Exposure Different Effects of a Novel Spider Venom-based Biopesticide on Survival, Gut Microbiome, and Transcriptome of Adult Worker Honeybees. So, without further ado, let's start by the introduction. So, biopesticides, which are derived from natural molecules of animals, plants, bacteria, or minerals, are considered a promising alternative to conventional chemical pesticides. A good example are proteins from insect antagonists, such as the neurotoxin from the venom of the spider of Segestria florentina, which it's already proved, and we proved this in the project that I told in EcoStack, that when it was fused with the bovine serum albuminum, was toxic for several species of pests, okay, that are important for crops. But our question was, okay, usually when we talk about biopesticides, we usually talk about a reduced environmental impact. But is this true? Does they really have a reduced environmental impact, or does we lack appropriate tools that are incorporated in the regulatory framework to prove that they have effect? And that was the main objective of this work, okay? We wanted to check if the, let's call it SFI6 BSA, if it had effects after acute and chronic oral exposure on adult worker honeybees, mainly in survival, gut microbiome, and transcriptome. This tiny piece of the work, this last one, is still ongoing. Okay, we are still processing the data, so I will not present it in this presentation. So all the work was developed here at the University of Coimbra, of the work that I'm going to present, okay, with the honeybees, because the part of producing the biopesticide was made in Italy and Newcastle, which were our partners. So the work was all developed in the University of Coimbra. So we took the honeybees from colonies of Apis mellifera iberiensis from our experimental apiary, and we performed the tests in our laboratory facilities. So basically, we had the 3 treatments, which were control, low, and high concentration, and we had groups of 30 bees, 3 replicates of 10 bees per each treatment. Like I said, this was part of a bigger project. We tested much more treatments, but then we selected this to do this part because we didn't have enough budget to do all the sequencing for all the treatments. So we selected these ones. The tests and the exposure and then the assessment for survival were made based on the OECD guidelines for the testing of chemicals, namely the acute and chronic oral exposure, okay, for honeybees. So after doing the exposure, after when we talk about acute testing and during and after exposure for the chronic, which lasted for 10 days, we did the survival assessment, and then we froze the bees at -80 to prepare them for the extraction of RNA or DNA, okay? So we froze them, then we did the extraction. We created 3 pools of 3 heads and 3 guts for treatment, okay? And then we used the Enzytec extraction kits, which is a really famous brand that you can use to extract the RNA or the DNA. And then what we did was that we sent them for sequencing at Novogen in the United Kingdom, which is a company that does this sequence at really good prices. And they were using an Illumina Novasec platform. After they send us the data, and we run that data through 2 different pipelines, one for RNA and another one for DNA, and compared it with the last version of the whole genome annotation for Apis mellifera, which in that time was AML HIV 3.1. Okay, so now going to the results. These ones are not still that preliminary, but I still didn't do more analysis. So yeah, so in terms of acute exposure, like I told you, we had 3 treatments. Acute exposure lasted for 4 hours, and then we did the assessment after that exposure, and then every 24 hours, okay, until it lasted the test because it needed to respect this threshold mortality of 10%, okay? So as you can observe, we had no significant mortality registered. We had one dead on the low concentration, but as you can observe, there is no clear gradation between the concentrations, which means that the increasing of the concentration did not cause more mortality. Now, going to the chronics lasted for 10 days, okay, of exposure, and we checked every 24 hours and renewed the food every 24 hours. But as you can see, we had also no mortality. And then we thought about, okay, we don't have mortality when we checked for the typical endpoints that are used for the pesticide regulation, but let's see if it has effects on another level, or if they have sublethal effects. And then we decided to do the so like I told before, so we did not have mortality neither for the high concentration or for the low concentration. Then we decided to go to see if we had sublethal effects, namely in the gut microbiome. And sorry, this was not working. Okay, yeah. So then what we did was that we started by checking if the alpha diversity, okay? And as you can see, for acute exposure, we did not see any statistical differences between the treatments for the 3 indexes that we analyzed, KO1, Shannon, and Simpson. But at chronic exposure, we did see effects for the Shannon and for the symptom indexes for both treatments where we applied the biopesticide. And in general, this can be explained because Shannon and Simpson indexes reflect changes in community structure, evenness or dominance, while KO1 focuses solely on richness, which means that if the treatment causes shifts in species relative abundances, like promoting certain species over others or redistributing dominance, but do not introduce or exclude species significantly, Shannon and Simpson will diverge between treatments, but KO1 will not. Okay? So, but on the other hand, if the rare species remain constant across treatments, KO1 also stay stable, okay? So, but this does not happen, which means that maybe we had not in the main species, but on the rare species of the gut microbiome bacteria, we had changes, okay? So now, going to the beta diversity, we did these really cool graphs, which I'm really proud, and we did the statistics, and what we observed was that neither for acute or chronic, we did not see any significant changes, okay? But what we saw, and it was really, when we were really happy with, was that we detected the four known core gut taxa, which are related with honeybees, which are Bifidobacterium, Lactobacillus, Snodgrassella, and Gilliamella. And we also detected some non-core members, which are usually associated with the honeybees, which are Bombella, Commensalibacter, and Bartonella. Okay, and then we ran another statistical tests to check if there was difference in the relative abundance of these taxa that I mentioned, but what we did not see any difference. Okay, so basically, just to conclude, what we observed on this tiny part without the transcriptome was that acute exposure to the biopesticide had no adverse effects on adult worker honeybees, this in terms of oral exposure, and chronic exposure caused no significant effects on survival, but on the other hand, induced changes on gut microbiome. And the idea that want to pass with this study is that omic approaches can be a really good tool to use with the regular testing procedures in order to see if really we have effect or not, and just not stay based on the survival, or which is typical the endpoints that are measured. So here I have the references for the work, and just to give a small for the ones that were not there. So we participated in ERB, which was the 10th version of this congress, which is one of the biggest congress of Europe. It took place in Tallinn between the 16th and the 19th of September, and it had around 350 participants from 43 countries, and we had 156 oral presentations and 163 posters. And I'm really proud to say, at the right, you have my poster, and to say that we won the Best Presentation Award for posters with our work, and this was not possible neither without the EcoStack project and the funding that we had for doing the study, but also without the BeSafeBeeHoney funding, which was really important for us to be present there. So I would also want to thank that, and thank you for paying attention to the presentation. If you have any doubts, please contact us. Thank you very much.
Thanks a lot, Artur, for your awesome presentation, and congratulations on that winning prize.
Thank you very much.
Yes, now, if anybody has any questions.
So, hello.
Yeah.
Yeah.
Thank you so much for your nice presentation and for your interesting work. Actually, I have a question for you because recently we have work that we used honeybee venom itself to test against Nosema cerana, which is a gut parasite of honeybee.
Yeah.
And actually, it showed very interesting results, and it killed the parasite, caused like 80% decline in. But we also found that it increased the level of hemocytes. So in
Okay.
your case, you use that
I-
in,
I'm
we can say, a similar venom or arthropod venom.
I'm sorry, yeah, but I did not. It was the
So
mic was
did you
cut.
hear me now?
Now I can listen. Sorry, but it cut it in the part where you said that it had effects on nosema.
Yeah, then
Uh,
but we also found an increase in the amount of hemocytes, which
okay,
is the blood cells of bees.
yeah,
So, and till now, I still trying to understand if it's like immune response or what's happened. But in
okay,
your case, you use a venom from a spider, and it
yeah,
looks like it's very safe. In our case, also, it doesn't cause any mortality.
okay.
So, are you planning also to use bee venom itself to see effects on the gut microbiome?
No. So this, like I started by saying, this was part of a bigger project, which was with other partners. In this
Yeah,
project, we tested regular, let's call it, okay, regular pesticides. Like we tested lambda cyhalothrin, alpha
yeah,
cypermethrin. So we tested, and then there was this part of the project. These colleagues from the University of Napoli and from the
yeah,
University of Newcastle that were working already with this venom, with this spider, and then
yeah.
they also included in the project for us to test. So no, and the project already ended, so I don't have
Okay.
the funds to do it. But
So,
yeah, but
yeah,
I'm still. What I can tell you is that I'm still going to do the transcriptome tests because this venom itself, the venom from the spider, is supposed to be neurotoxic, and that is already published on the paper. So I don't expect it to have the effects, for example, on the hemolymph, but it can have. I will not study it.
yeah, because it's still a protein. It will be digested.
Yeah, yes,
Yeah.
yeah.
Oh, okay. So, and the end message of your work is that this novel biopesticide is safe for bees because it will
No,
be used against pests.
no. No, the message is that we clearly need not to focus only on the to start little endpoints,
Yeah.
and that we should increase the way that we do the risk assessment to make it better in by including another tools. Okay,
Like, for
what I
example,
saw,
behavioral test.
for example, behavioral tests can also be.
Yeah.
I'm actually working on it right now with
Yeah.
in different species of bees, which is also a problem because we usually only test honeybees. But I'm doing
Yeah.
kind of building a protocol to kind of make a standardized test because you can observe it, but the there's no standardized test for behavioral testing
Yes.
still.
Yes. Of course.
So yeah, I'm working on that. Yes, but my message is not that this is safe.
Still,
Okay.
until now, it looks like it's very limited effects. Yeah,
Yeah.
some changes in alpha diversity, but almost nothing, right?
Yeah, true, true. But I'm
Okay,
really now
but it's
focusing
still not enough to conclude.
on the
Yeah,
brain. I'm going
yeah,
to focus on the brain because I did not do any behavioral tests, but I really want to check if it has effects on the brain as it should because it's neurotoxic.
yeah.
Okay,
Thank you
thank you.
so much. Yeah,
Thank you very
all the
much.
best.
Violeta,
Hi, hello, can you hear me?
Yes.
perfectly.
Yes, thank you for nice presentation. I went also in EuroBee and I saw you with the group. But my question is, in the case that experiment gives the data that is everything is possible to do with that new pesticides, what will be tool to produce such a huge amount of the spider proteins and also connection with the cattle serum? It's much easier, but will be that part of the like genetically modified production of the what will be sourced for such an enormous amount of the spider protein? Thank you.
I I'm really, really sorry, but I don't have the answer for that part. So I did not participate in the production. That was Newcastle and Napoli. but I can give you the contacts and you can ask. I'm really sorry, but
Thank you. I just was curious. So
yeah,
thank you.
but I get where you're getting from. But then maybe they can produce something synthetic from it, like we do with the pesticides. But we started by this because also that's like the idea that is behind the biopesticides, because when people hear biopesticides they usually tend not to be so afraid. So I think that is also what is the idea from. But yeah, sorry, I cannot give you the answer, but thank you very much for your comment.
Thanks a lot for your
Thank
presentation,
you,
Arthur.
thank you
We
for
can
inviting
have
us.
now Soraia to give us her presentation. Soraia, I'm not sure. Can you hear us?
Yes, can you
Yes.
hear me?
Yes, yes.
Yes, okay. So I'm trying to share my... Can you see my full window?
Yes, we can see your...
Yes? Okay, perfect. So my name is Soraia Santos and I'm a research fellow at University of Coimbra. And today I will present you a topic that relates behavior and communication of honeybees with their dietary preferences and requirements, entitled Exploring the Summer Foraging Patterns of Honeybees in Agriculture Landscapes. So, yeah, it's changing, right?
Yes.
Yes.
We just see
So,
your
honeybees,
PowerPoint window. Is it on full screen on your side?
is it on full screen? Yeah.
Okay.
But can you, what you see,
It's visible. We just see... Oh, no, now it's much better.
better now?
Yes.
But sorry, now it's really small.
Well,
Okay, I don't know what's happening. And now, better? I don't know what I did.
It's much better, yeah.
Yeah, okay. So, honeybees are, I changed my, is this one? So, honeybees are social insects with high ecological and economical importance, playing a crucial role in ecosystems by sustaining biodiversity and providing pollination for a wide variety of crops and wild plants. Fragmentation of natural habitats in intensive agriculture systems has led to a significant decline in the abundance and quality of the food sources for honeybees and other pollinators. This can affect honeybees' diet and consequently affect colony reserves and development. Ideally, floral patches near the hive allow bees to access different food sources over extended periods. With landscape dynamics, bees need to adapt and adapt their forager decisions. Vocal dance communication is a most common communication to simplify this system. So, this type of communication helps the bees to efficiently exploit and conserving time and energy. This type of communication is usually made by forager honeybees, and the distance is translated by the vocal duration and the direction angle towards the sun. As I said before, this behavior is crucial to identify the main foraging locations of honeybees, allowing the hive to exploit the resources efficiently, but also provide us with information about their preferences and dietary needs, while also serving as an indicator of the most profitable resources. In this sense, this study aimed to evaluate the effects of temporal and spatial variation of flower resources on the foraging patterns of honeybees in an intensive landscape in Spain. So this work can be divided in two main phases. The first one was in the field during the summer of 2018, and an apiary and a meteorological station were installed in this region in Burgos. One of these beehives was a modified Langstroth hive for the observation and the Woggle dance video recorded. Besides that, we also register all floral resource abundance and availability assessment. And that we define eight land cover types, such as forest, fruit trees, unproductive areas, grasslands with trees, grasslands with shrubs, grasslands, hedgerows, and water surfaces. In the second phase, in Coimbra, Portugal, we identified the vocal dance and we analyzed all the recorded videos and registered the day and hour for further analysis. In this analysis, I would like to emphasize that was only dances with a minimum of 6 vocal runs without any interruption were considered for the analysis. To translate the communication, the durations of the dances were measured using this software, and the angle was measured manually. The floral resource distribution in the landscape was analyzed using QGIS to verify eye resources and for conservation. Finally, the foraging patterns were defined by using the same software, but I would like also to emphasize that the days chosen for representing each month were selected according to the registered favorable weather conditions and the quality of the recorded videos. In our results, we can see the foraging pattern, and sorry, I don't know if you can hear me. That I'm with the sign that. Can you hear me?
Yeah.
Yes.
Everything is all right. Right? Keep going.
Okay, thank you. So first we can see the foraging patterns. In a figure A, we can see the orientation for different months, and in B, C, and D, the relative frequency of the flight distance obtained from the Woggle density coding in the 3 months, and in E, F, and G, the foraging location direction frequency for the 3 months. Here, I think it's really small, but is the orientation north, south, and east, and oest, and we can see the different directions. So when we see all this information together, we can see that landscapes change significantly during the summer and that honeybee behavior change as a result, which allow us to identify the most attractive areas for honeybees over this time. We also observed that the number of dances performed in June was considerably higher when compared with the remaining months, and the preferences were mainly the hedgerows and legumes. Besides that, there was also an evidence of a reduced number of dances being performed in response to the dynamic of the landscape in August with the blooming of the sunflower here. And then, so overall, spatial and temporal variations in flower availability influenced honey woggle dances during the summer season. And hedgerows were the most important landscape type during the summer, with the highest number of decoded vocal dens in June and July, but also one of the top 5 visited areas in August. This may be an indication that colony identifies resource hotspots in landscape and rates these plants as more profitable, thus generating more densities so that more individuals use the resources. In addition, in August, there is a less diversity of food resources for the honeybees, and agricultural landscapes rich in nectar and pollen, such as the sunflower fields, are particularly attractive when they are the main food source, as found is the found ones that we found in the studied area. In this sense, there is a possibility that the bees perform fewer dances to, for example, to save time and energy as a necessary effort to communicate. So in conclusion, I think it's important to understand a little bit of everything that I said, and honeybees are essential to ecosystem and agriculture, but are challenged by declining the floral diversity. They adapt their foraging to available resources, but their nutritional needs are still not fully understood. To support honeybee health and sustainability, it's important to continue with these studies that explore behavior and communication and related with their food requirements or diet requirements to encourage bee-friendly practices and being crucial for both honeybees and the ecosystem that they sustain, they support. Thank you for your attention. Here I put a little bit of my poster from the same congress that my colleagues. And thank you. And if you have any question.
Thanks a lot, Soraya, for your presentation. It was really nice. If anybody has questions, there was a question one in the comments. It's for Serbian one, Serbian comment, but yeah, this YouTube channel is BeSafeBeeHoney. I'm not sure if it's up yet, but it will be in the future if it's not now. And
Yes.
I might have a question, if I can.
Okay.
yeah, sure.
Yeah, sure.
So I was wondering, how did you identify the field information about the agricultural fields, like the layers, like the location of the sunflowers field, legumes? Did you map it by just field
Yes.
visits?
There were field visits, but also we use maps that are available online from the government of Spain, the Centro de Descargas del Centro Nacional de Información Geográfica, and another one, there is an institute in Castilla y León to sustain our study.
Okay, thank you very much.
You're welcome.
Hello.
I don't know if anyone. Yes?
Yes, we can hear you, Željko.
Yes.
We can hear you, but very poorly.
Can't hear you at all.
I'm sorry,
Yeah.
I can't hear.
Yes, we can't understand you, not nor hear you.
I hear something, but it's.
Maybe you can write down a message and we can answer your question. No. I'm not sure if.
Maybe you can write it on the chat and then we can read it from there.
Yes. Željko, can you hear us? No, I'm not sure it's possible. Maybe he doesn't hear us. I suppose he can write his questions on the email. I don't think Željko actually has a question. He's speaking about something else. Okay, I suppose that's it, guys, for today. Thanks a lot for giving us your presentation, and hope to see you again very soon in some conference or some meeting for our project BeSafeBeeHoney. Okay, I'll stop the recording now. So thanks a lot, and I'll send you again some link for the webinars, and I hope they will work much better.